Hi there. In this video, we are going to discuss about minor hemoglobins. Minor hemoglobins. Okay. So, what are minor hemoglobins? The discussion we previously had about hemoglobin was about hemoglobin A. All the discussion we previously had was about hemoglobin A, which is the major hemoglobin, or which is the major hemoglobin found in adults. Major hemoglobin of adults. Okay. So HbA is the major hemoglobin found in adults. So what are the other hemoglobins? There are other variants of hemoglobin. The other variants of hemoglobin include HbA2, which is also found in adults. HbA2 also found in adults, and then there is HbF, which is fetal hemoglobin. It is fetal hemoglobin, and there is a variant HbA1c. HbA1c, and there is also a hemoglobin termed as HbGover 1. HbGover 1. So these are the different classes of hemoglobin, different types of hemoglobin, and let's discuss about their composition. When we discuss about HbA1, it is composed of 4 chains, 2 alpha chain and 2 beta chain, and in adults its amount is about 90%. In adults, about 90% fraction of hemoglobin is HbA. Now come toward HbA2. HbA2 is composed of 2 alpha chain and 2 delta chain, and in adults its total fraction of it contributes to the total hemoglobin by 2 to 5%. And then come toward the fetal hemoglobin HbF. So HbF is composed of 2 alpha chain and 2 gamma chain, and in adults its percentage is less than 2%. In adults its percentage is less than 2%. Next we come towards HbA1c. HbA1c is variable in adults depending upon whether they are healthy or not. In adults, normal adults, HbA1c is less than 5.7%. Okay. However, if HbA1c is between 5.7% to 6.4%, then person is said to be pre-diabetic. And if it is greater than 6.4, then person is diabetic, a person has diabetes. So HbA1c is basically an indicator of diabetes. And HbGover1, it is composed of 2 zeta chain, 2 zeta chain and 2 epsilon chain, So it is basically a combination of zeta chain and epsilon chain. So HbA is major hemoglobin found in adults, and HbA2 is also found in adults in 2 to 5%, and HbF is fetal hemoglobin. It is principally found in fetus. In adults its percentage is less than 2%. HbA1c is basically the glycosylated form of hemoglobin to which glucose has been attached, and glucose usually attaches at the amino terminal of hemoglobin. It usually attaches to beta globulin chains of HbA1, at amino terminal, and amino acid is valine. So glucose in diabetics, glucose is increased, which joins to the beta globulin chains, valine of the beta globulin chains at amino terminal, and produces this glycosylated hemoglobin, and it is non-enzymatic glycosylation. And increased amount of HbA1c shows increased amount of blood sugar level. So it is an indicator of blood sugar levels. Okay, now we come towards their relative duration. We have discussed their structure here. Now let's come toward their relative distribution. Okay, we have discussed HbA1c. It is let's remove it from here. HbA1c. We first discuss HbGover1. HB Gover 1. So this hemoglobin is principally synthesized by embryonic. It has two epsilon, it has two zeta chain and two epsilon chain, and it is synthesized by embryonic yolk sac. It is synthesized by embryonic yolk sac after conception, and its synthesis continues up to 5th week of gestation. Okay, it synthesis starts after first month after conception and continues, and in 5th week of gestation, what happens? The synthesis of hemoglobin goes to the livers. Initially it is synthesized by embryonic yolk sac. In 5th week, liver replaces the synthesis of hemoglobin, and then this synthesis goes to the bone marrow. Then bone marrow starts synthesizing the hemoglobin. So the hemoglobin synthesized by bone marrow in fifth week of gestation is the HbF. So in 5th week of gestation, the hemoglobin being synthesized is HbF, and this HbF contains alpha and gamma chains. And when the baby is born, he has about 60% of fetal hemoglobin. HbA synthesis, which is the chief component of adult hemoglobin alpha 2 beta 2, its synthesis starts in 8th month of pregnancy. So that's why when a baby is born, about 60% hemoglobin is fetal hemoglobin, and the rest is in rest hemoglobin major component is HbA1. However, this HbA then replaces this fetal hemoglobin. That's why adults have fetal hemoglobin less than 2%, and HbA is greater than about 90%. Okay, so the cells that contain fetal hemoglobin in adults are called F cells. So what is going on? After conception, the hemoglobin formed by embryonic yolk sac is HB Gover 1. In 5th week, liver starts synthesizing hemoglobin, and then bone marrow replaces it, and the hemoglobin synthesized here is HbF or fetal hemoglobin, which contains alpha 2 and gamma 2 chains. And then 8th month of gestation, HbA1 starts to be synthesized, and when fetus is born, he has 60% of fetal hemoglobin, and the rest is about rest of the hemoglobin contain major amount of HbA. Then the HbA synthesis continues. However, HbF synthesis declines. That's why adults have HbA about 90%, and fetal hemoglobin is less than 2%. Okay. So HbA2 synthesis start before birth, and it also continue to be synthesized. However, its amount is very little, so it is synthesized in small amount. That's why it constitute to about 2 to 5%. So if we represent it by a graph here, So among alpha chain, there was alpha chain like zeta chain. So zeta chain after conception is synthesized, then its synthesis declines, and then the alpha chain synthesis starts, and alpha chain synthesis continue to increase and then attain the plateau phase. So this is alpha chain, and this is zeta chain. And if we come towards beta globulin groups, so among beta globulin groups, initially there is epsilon chain of HB Gore 1 which constantly declines, then it is replaced by gamma chain of fetal hemoglobin. Gamma chain stays here for the short time, then gamma chain declines, and then it is replaced by beta chain. Okay, so this is epsilon chain which declines, then gamma chain which also declines, then beta chain which replaces it and forms HbA1. So this is the graphical representation of different hemoglobin chains. So next we come towards the their interaction of fetal hemoglobin with 2,3 bisphosphoglycerate. We previously discussed in the oxygen hemoglobin dissociation curve, that 2,3 bisphosphoglycerate shift this curve towards the right side, so it decreases the affinity and improves the unloading of oxygen. However, if this 2,3 bisphosphoglycerate is absent, then there is left shift and increased affinity and difficult loading. So 2,3 bisphosphoglycerate decreases the affinity and improves unloading of hemoglobin. However, in the HbF, the binding of 2,3 bisphosphoglycerate and HbF is weak, because at the binding site, HbF lacks some positive charge amino acid. It lacks positive charged amino acids because the 2,3 bisphosphoglycerate joins to the positive charged amino acid of beta globulin chains because negative charge phosphate is attracted to positive amino acids here. However, HbF has less positive charged amino acid as compared to HbA. So HbA binds more to 3 bisphosphoglycerate. So HbA has less affinity for oxygen. So HbA delivers oxygen better, or you can say it unloads oxygen better. However, HbF binds weakly to 2,3 bisphosphoglycerate, so it unloads with difficulty. However, if there is no 2,3 bisphosphoglycerate, both HbA and HbA have same oxygen affinity. So difference in oxygen affinity of HbA and HbF is due to 2,3 bisphosphoglycerate. HbF has greater affinity and difficult unloading. HbF has less affinity and better unloading. So this difference of oxygen affinity helps HbF to transport oxygen across the placenta in the fetus. So it was about the different kinds of hemoglobin. Thank you.